This tutorial will focus on X inactivation and Barr bodies. Essentially in organisms that carry, where the females carry two X chromosomes, that one of those X chromosomes is randomly inactivated in all of the cells. So in a cell that has two X chromosomes, one of the X's is inactive. It's inactivated by when it's produced. It's inactivated by the fact that it's packaged in such a way so that transcription cannot occur along its length. So the X chromosome that's inactivated gets wrapped in something, ends up in a structure called heterochromatin. So it gets packaged with this into this heterochromatin, which means it's transcriptionally inactive. It can't carry out transcription. These X chromosomes that are made inactive are more condensed than the active chromosomes, so they kind of package up so that they're kind of more bunched together and tighter, and then they move to the periphery or the edges of the nucleus. And so when the nucleus is stained, often they appear as these dark staining bodies on the edge of the nucleus, and they were discovered by a scientist, Barr, so they're named Barr bodies, B-A-R-R. So when you see a Barr body like this on the edge of a nucleus, then you know you've got one inactive X chromosome. The other chromosome will be active. In some rare cases where there are mutations, some individuals may end up with 3 copies of the X chromosome. In this case, you will see 2 Barr bodies. So there's only ever one active X chromosome, and anything in excess will be inactivated. So you may see 2 Barr bodies in rare circumstances. In some males, occasionally, you'll get 2 X's and a Y, in which case you'll see 1 Barr body in such a male. Normally in males with an X and a Y, you wouldn't see any Barr bodies because there's only 1 copy of each chromosome. Now, these Barr bodies or X inactivation sometimes plays itself out in phenotypes which are quite distinct, and one of the classic examples is the colour of the tortoiseshell, fur of the tortoiseshell cat. And you get this patchwork kind of effect in the cat, as shown here, light patches and dark patches, and this is determined by which of the X chromosomes happens to be active. This is a case where a particular gene, in this case, shows up a visual phenotype in terms of the fur. This doesn't always happen. Many of the genes along the X chromosome don't always show up as something as clear and visual as this. So depending on which chromosome is active, you get a patch. You might get a patch of light fur, or you might get a patch of dark fur, and you get this tortoiseshell effect. So in some species you'll see this visual effect of this inactivation of the chromosome.